All right, welcome everyone. Apologize, things went a little bit beyond our control today in terms of getting the PowerPoint up and not being able to take the quiz, but we're still gonna let the learning process roll on, and we'll break down the PowerPoint and the lecture this way. So today we're doing chapters 3 and 5. So chapter 3 is cells, the fundamental unit of life. So everything living is made up of cells, and they vary in size, shape, and function. So in this chapter, we'll look at their structure. We'll do a little bit more on organelles. I know we did some in class today. And the cell cycle, and eventually cell death. Okay, so cells. As I just said, they're the fundamental structure and functional units of life. Every living organism, from the simplest bacterium to the most complex human, is composed of cells. And they vary greatly, as we can see here, in size, shape, and function. But they all share characteristics that allow them to maintain life process. And throughout this chapter, we will examine the anatomy of a typical cell, the function of the major components, and the process by which cells grow, divide, and ultimately die. So understanding cells is critical to understanding anatomy and physiology because all tissues, organs, and organ systems, everything we're going to cover after this, are built upon these microscopic units. Okay, so the composite cell. There is no such thing as a typical cell, since each cell type is highly specialized for its own particular function. However, a composite cell model helps us visualize the structure that most cells have in common. These include three major parts: the nucleus, cytoplasm, and the cell or plasma membrane. The nucleus contains genetic material and serves as the command center, directing all cellular activities through gene expression. The cytoplasm is the region between the nucleus and the cell membrane, and it's filled with cytosol and organelles that perform very diverse metabolic tasks. And finally, the plasma membrane forms the outer boundary, maintaining structural integrity while selectively allowing substances to enter or leave the cell. This selective permeability is essential for maintaining homeostasis as it regulates nutrient uptake, waste removal, and communication with other cells. Okay, the cell membrane structure. The cell membrane, which is often called the plasma membrane, is a dynamic and it's a flexible structure that defines the cell's outer boundary. It is composed primarily of phospholipid of a phospholipid bilayer interspersed with proteins, cholesterol, and carbohydrates. The hydrophobic interior acts as a barrier to water-soluble substances, while membrane proteins serve critical functions such as transport, signaling, and cell recognition. Some proteins act as channels that allow ions to pass through, while others function as receptors that respond to hormones or neurotransmitters. The fluid mosaic model describes this membrane as flexible and self-healing, with components capable to move laterally within the layer. The selective permeability of the membrane ensures that essential substance can enter while harmful or unnecessary materials are kept out. This selective control is vital for maintaining the internal environment of the cell, also known as the uh, intracellular part. So let's take a second to go through some of these proteins. In this, we have the channel protein right in the middle. Okay, and this protein basically acts as a doorway for certain molecules like waters and ions. Let some in, some out. The peripheral protein, okay, and this helps with support and signaling. There is a receptor protein, and this protein receives messages from outside the cell, like hormones. Okay, then these signals, these messages come in and tell the cell what to do. There is on the membrane a cell identity marker, and this is really helpful for your immune system to know if this is your cell or if it's an invader cell. You don't want to attack yourself. There's also carbohydrate chains of the green dots on the top of the cell membrane, and they are attached to proteins or lipids, and they help the cells recognize each other. And finally, we have cholesterol, which is between the top of the cell membrane and the body, and these are there to keep the membrane from getting too stiff or too gooey. They aid in proper flexibility. Okay, and then finally at the bottom here, we have the cytoskeleton. Okay, and as we discussed in class, this helps, it's a protein network that helps the cell maintain proper shape and structure. It's like the cell's skeleton. Okay, organelles that occupy the cytoplasm. So the cytoplasm refers to all material inside the cell membrane but outside the nucleus. It includes the cytosol, a semi-fluid substance rich in dissolved nutrients, ions, and enzymes, as well as various organelles. Organelles are specialized structures, each performing a specific function that can contribute to the overall metabolism and survival of the cell. For example, the endoplasmic reticulum synthesizes molecules, the Golgi apparatus processes and packages them, and the mitochondria generates energy. So the cytoskeleton, which is composed of microfilaments, microtubules, and intermittent filaments, which we'll talk about on their own slide, provides internal support and facilitates intracellular transport. The cytoplasm, therefore, is not just a filler material, but is a highly organized environment where countless biochemical reactions occur simultaneously, ensuring the cell's vitality. All right, so the nucleus. We're going to go over these briefly because I know we did them in class, but I still want to get some words on film about them. So the nucleus is the control center of the cell, containing the cell's genetic information in the form of DNA. It is enclosed by a double-layered nuclear envelope that separates it from cytoplasm. And then embedded in this envelope are nuclear pores that regulate the movement in and out, so they allow it to come in and out, of molecules such as RNA and proteins. Within the nucleus, the nucleolus is a dense region where ribosomal RNA is synthesized and ribosomes are made. So the DNA is organized into chromatin, a combination of DNA and protein that condenses into chromosomes during cell division. The nucleus not only stores genetic material, but also manages the expression of genes. Therefore, it dictates which proteins are produced and when. This control over protein synthesis is, is really what allows cells to specialize and perform their specific physiological functions. Okay, ribosomes and the ER, the endoplasmic reticulum. So ribosomes are small granules composed of RNA and protein. They serve as the sites of protein synthesis. They can exist freely in the cytoplasm, where they produce proteins used within the cell, or they may attach to the rough endoplasmic reticulum, forming a structure specialized for the synthesis of proteins, which is then destined for secretion or for incorporation into cell membranes. The endoplasmic reticulum itself is a vast network of membranes that serves as a transport system. So the rough ER, which has ribosomes, is primarily involved in protein synthesis, while the smooth ER that does not have ribosomes is responsible for lipid synthesis and the detoxification process. These two regions work together to ensure the proper production and modification of macromolecules necessary for cell function. Okay, next we got vesicles and Golgi apparatus. Now this one I don't believe we covered in class. So vesicles, these guys are small membrane-bound sacs that transport materials throughout the cell. They shuttle proteins and lipids from one organelle to another, particularly between the endoplasmic reticulum and the Golgi apparatus. So the Golgi apparatus, which is often likened to a cellular post office, consists of these flattened, in the picture, purple membrane sacs that refine, package, and direct proteins to their proper destination. They modify proteins by adding carbohydrates or phosphates, creating glycoproteins or phospholipids. Remember, phospholipids is what the entire cell membrane, well, not the entire cell membrane, but most of the cell membrane is made from. So once packaged in the vesicles, these products may be secreted from the cell via exocytosis or sent to lysosomes for use within the cell. The efficient functioning of the Golgi apparatus is critical for maintaining the flow of materials that the cell uses for growth, repair, and communication. Okay, so closed-in pictures of the Golgi apparatus and the vesicles. Okay, mitochondria, lysosomes, and peroxisomes. So mitochondria is often referred to as the powerhouse of the cell because they generate ATP through cellular respiration. They're double membrane. They have a double membrane, and their double membrane allows for the separation of different metabolic processes, and their inner folds, called cristae, increase surface area for energy production. So lysosomes, now these are small vesicles containing digestive enzymes that break down worn out organelles, pathogens, or cellular debris. These are essential for serving as the cell's waste disposal system. We all need to get rid of waste. So this is where these come in. Peroxisomes, these are similar in structure to lysosomes. They contain enzymes that break down fatty acids, and they also detoxify harmful substances like hydrogen peroxide and also alcohol. Together, these organelles maintain cellular health by managing the energy of the cell, how the waste is removed in the cell, and the detoxification process. Okay. All right. The cytoskeleton. Now this is a excuse me, complex network of protein filaments that provide structural support. They maintain cell shape and they help facilitate movement. The cytoskeleton includes microfilaments made of actin. We'll talk much more about actin when we talk about the muscles. They also contain microtubules composed of tubulin and intermediate filaments. Now, microfilaments are the smallest, contributing to cellular movement and contraction, especially in muscle cells. Microtubules are the largest, and they form a network of cilia, flagella, and centrioles, while the intermediate filaments provide tensile strength to the cell. This internal scaffolding is dynamic, and it constantly is reorganizing as the cell moves. It's constantly also dividing and changing shape as necessary. So beyond structure, the cytoskeleton also plays a role in intracellular transport. It helps guide organelles and vesicles along specific pathways, basically like a microscopic highway system. So there's a lot going on here. And as we talked about in class, it is not a piece or two here and there. It is very dense and it is constantly moving. All right, the centrosome. The centrosome, which is located near the nucleus, it is consisting of two cylindrical centrioles made of microtubules. These structures are essential during cell division, which we're going to talk about here, I think in a slide or two. The centrioles organize the spindle fibers, and they attach the chromosomes and ensure their proper segregation during mitosis. Without a functioning centrosome, cells would be unable to distribute genetic material evenly, leading to errors in cell division. In non-dividing cells, the centrosome still plays a role in maintaining the organization of the cytoskeleton and in forming the basis of both cilia and flagella. So the centrosome's strategic position near the nucleus and its structural organization make it the central to the process of mitosis and overall cell architecture. Okay. Other structures like cilia and flagella. So these are extensions of the cell membrane that allow the cell to move. So they're important for cell movement. Cilia are numerous and short, beating in coordinated wave to move fluids across cell surfaces. Anytime we're going to see cilia, they're helping move something along. For example, the mucus in the respiratory tract is an example of this movement. Flagella, in contrast, are much longer, and usually there's only one, so they're singular. They help and propel the entire cell through its environment. In humans, the sperm cell's tail is the only example of this flagella. Okay, so both structures are made up of microtubules arranging in a 9x2 pattern surrounded by a plasma membrane. Their coordinated motion, it will depend on the ATP supplied by the mitochondria, demonstrating how, and this is an example of demonstrating how organelles work together to support complex cellular functions. Let's move on to the cell cycle. So that is the cell, those are the organelles, that's the cytoplasm, that's the cell membrane, nucleus, all that stuff. So let's move on to the cell cycle. Okay, and the cell cycle encompasses all the stages a cell undergoes from its formation to its division into two daughter cells. It consists of interphase, when the cell performs its normal functions and prepares for division, and the mitotic phase, when actual division occurs. The cell cycle ensures that the cells grow, they replicate their DNA accurately, they divide properly, and they maintain tissue structure and function. Regulation of the cell cycle is tightly controlled by certain checkpoints that we'll look at, and certain proteins called cyclins. Errors in these regulatory mechanisms can result in uncontrolled cell division, leading, unfortunately, to a word nobody likes, cancer. So, understanding the cell cycle is vital for comprehending growth, repair, and disease. Okay, so here's a picture of the cell cycle, and we're going to break it all down, but come back to this slide when you're studying if you want to talk your way through it to see if you got a good understanding. Okay, so the first one here is interphase. Okay, an interphase, this is not a resting phase, as scientists once believed, but rather it is the most active period in a cell's life. So during this phase, the cell grows, carries out normal metabolic activities, and prepares for division. It is divided into three phases, G1, S, and G2. Okay, so it's those three. During G1, the cell increases its size and produces necessary proteins that it's going to need for this process. During the S phase, the DNA is replicated so that each daughter cell will receive an identical copy. And then during G2, final preparations are made for mitosis to occur, including organelle replication here. So this stage ensures that the cell has the resources and genetic material required for successful division. All right, let's move on to mitosis. So mitosis is the process of nuclear division that produces two genetically identical daughter cells. Okay, it occurs in four distinct phases. In prophase, chromatin condenses into visible chromosomes, and the nuclear envelope begins to break down. Okay, that's prophase. During metaphase, the chromosomes align at the cell's equator, attached to spindle fibers extending from the centrioles. Okay, so that's metaphase. Then anaphase follows as sister chromatins separate. they move to the opposite poles of the cell, so opposite sides of the cell during anaphase. In the final phase, telophase, brand new nuclear envelopes form around each set of chromosomes. And the chromosomes begin to unwind back into chromatins. Mitosis ensures that each daughter cell receives a complete and identical set of genetic instructions. Now that is what happens when it all works perfectly. We have two cells. Okay, study guide. I know my face is in the way, so you might want to look at this on your own slide, but study guide for these major events in the mitosis. As well as we should call this a study guide too, because this is a visual to everything we just talked about. So if we look at this, we'll start with early interphase. The cell is growing and performs normal functions. Chromatin is uncondensed and centrosomes and other organelles are present. And we have a restriction checkpoint. The cell checks for DNA damage, nutrients, and size to decide, if it should continue the cycle. And then begins the S phase. In here, DNA replication occurs. Each chromosome now consists of two sister chromatids, moving into late interphase, the G2 phase. And this is final preparations for mitosis. In here, centrioles replicate, and organelles and mitochondria increase in number. So now we're going to green in the picture. And this is start of mitosis. And we're going to start with prophase. So chromosomes here condense and become visible. The nuclear envelope begins to break down. And spindle fibers start to form. And we have late prophase, where the chromosomes are fully condensed. The sister chromatins are connected by centromeres. Centromeres? That didn't even sound right coming off. And spindle fibers attached to centromeres. Metaphase. Okay, here the chromosomes align at the metaphase plate, the equator of the cell. And then anaphase, these center chromatins are pulled apart to opposite poles by spindle fibers. And then finally, telophase. Nuclear envelopes reform and around each set of chromosomes, and the chromosomes decondense. Okay? The cytoplasm divides, forming two daughter cells. Now we have two cells from one cell. All right, cytokinesis. Now this is the division of the cytoplasm, completing the process of cell division. Cytokinesis begins during late anaphase, and it continues through telophase. In here, a contractile ring composed of actin filaments forms around the cell's midsection. This continues to tighten until the cell splits into two separate daughter cells. This constriction, known as cleavage furrow, marks the physical separation of the two new cells. So although they contain identical DNA, the daughter cells may differ slightly in size or the number of specific organelles. And this may be balanced during subsequent growth. So cytokinesis ensures that each cell is fully equipped to function independently after cell division. Last slide. Cells have a life, right? Just like anything, and then they will eventually die. Anything living will eventually die. And cell death is no difference. In fact, this is an integral part of life. There are two major forms here, apoptosis and necrosis. Apoptosis is a programmed, okay, it is orderly process in which the cells self-destruct when they are no longer needed or they are damaged beyond repair. So this mechanism is crucial during development. For example, it removes webbing between fetal fingers and toes. And it also helps maintain tissue health by eliminating potential harmful cells. Necrosis, in contrast, is unplanned cell death resulting from injury or disease. This leads to inflammation and tissue damage. Understanding apoptosis and necrosis allows us to appreciate really how the body maintains balance and how it responds to injury on a cellular level. Okay, so that's the chapter, guys. That's chapter 3 on cells. So that's half of this week. I'm going to put together another one for chapter 5, tissues. So when you're done with this one, head on over to the next one.